Okay, another piece of housekeeping. How many of you got the email for the midterm instructor reviews? Should be all of you. If you haven't, just double check, make sure that you've got it. So it's not required, but I will strongly encourage it. As I've mentioned, this is my first time teaching this course, so you are all the guinea pigs. So I would appreciate any feedback, good or bad. And it's just one question. And please wrap up your conversations. Class has begun. Unless you want me to ask you to share your conversation with the class. Okay, so there's that. There's an opportunity to review. About 25 people have done it so far. That is due next Monday, I think at midnight. And I just went with the university recommendation there. I will also mention there is a Rate My Prof page for me. I would normally not mention this, but there is another David Simpson at the U of A, and he's the only one who comes up if you Google David Simpson Rate My Prof. He's in the English department. So you actually have to go into Rate My Prof to find me. David is a very common name, so there's a lot of David Simpsons. So make sure it's David Simpson, Psychology, Rate My Prof. You know, I don't want the David Simpson in English stealing the good reviews or the bad reviews, right? He's gonna look and it's like, oh, he wears shorts, he's crazy or whatever, and then he'll be very confused. So I mentioned that just in passing. That obviously you can do any time, but the review that obviously is more important is the one that the U of A has provided, and as I say, that is due next Monday. So strongly encourage you to do that. Also, hopefully all of you saw that the test marks are up. Both written and the multiple choice. The grades were a little lower than last time, which I'm not that surprised by. It was the hardest chapter, chapter 3. There's a lot more chatter this time. Did something happen? Or is it this? Maybe I should just put that back on after I'm done saying what I'm saying. I'll mention, some of you spelled the brain areas wrong in the written. It was in the question. So, you know, I know some people have bad memories, but really, like the second that, you know, some people forget. I was very tempted to take marks off, but I decided to be nice. Just copy it off the question. That's all I have to say about that. Okay, that's all the housekeeping I have to say. So today we will be talking about the APA assignment. Okay, I got quiet after that. So that is due next Thursday. Next Thursday at 11:59 p.m. So we'll spend the first 20-ish minutes of class talking about what I expect for the APA assignment. It should be easy marks if you just follow the template, which I will show you in a second. I will also take this time to use that APA discussion as a springboard to do something a little unusual. I'm going to say a little bit about what getting an education in psychology looks like for any of you who are majoring in psychology, or really any of the social sciences, like sociology, criminology, what the kind of steps are. And I'm doing this because no one did it for me in undergrad. I kind of had to piece it together. So this may be useful information for many of you. If you're majoring in something else or going to med school, it may be, or whatever, it may be less relevant. But I'll spend a little bit of time on that, and then we will get started on chapter 5. Cool ringtone. Okay. So if you go to the syllabus, there is a section on the APA assignment. Make sure you follow the instructions there. So as it says here, it is due at 11:59 p.m. on next Thursday. As I also say, upload it early. Don't wait for 11:55 or 57 to upload it. Upload it early to avoid being late because of a technical issue. For every day it's late, 1% off. And if it's 11:01 a.m., that's late. So I'll just keep it simple. So upload early. Just avoid. I don't want sad emails like, Mr. Simpson, I swear I was uploading at 11:55. I don't feel like getting lots of those. So just upload it early and avoid having a technical issue like that. This document here, the APA assignment template, is your best friend. This will be very helpful. Just download it and edit it. Don't even bother making a new document. Just use this as your starting place. So this is what the title page should look like. If you double click up here, you can edit the running head, which should be in all caps like this, and it should be the same as the title here. The title should not be exactly the same as the title of the paper you choose. It should be just kind of a short, condensed summary of that. I'll talk in a second about how to find an article to use. And for this, you're just gonna write 1 to 2, and in fact, I'd encourage you just to write 1, just make it easier for me. There's a lot of students in this class. Just summarizing the academic psychology journal article that you choose. I'm not looking for deep insight or statistical prowess or beautiful prose. Just write something that makes sense so that I can walk away with a rough understanding of what the article does. You can probably get most of the information you need for this from the abstract. You do not yet have, most of you, the ability to read a statistics results section. You don't know what a t-test is or anything like that. So I'm not expecting that level of sophistication. I just want you to summarize the basic findings of the paper. You should include at least 1 citation of it. So, and you are expected to use APA style. So if it's 1 author, you say it like this. So height, that's the last name, in brackets, 2001, whatever the year is, said blah, blah, blah. Or you can say the study found blah, 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 height, 2001. And then I have instructions for if it's 2 authors or 3 or more. So just do what it says there. As I said on the first day of class, I strongly discourage creativity. No creativity. So just do what this says. And then for the references, this is probably where the most marks will be lost. I'm going to be very picky about this. And that's a gift to you. This template is a gift to you because if you get clear on APA style now, your future psychology classes will be easier. So every little bit matters, right? The fact that this, for example, the fact that this number is italicized and this one isn't, that could be marks off. And yes, I will be that picky about it. This is a gift to you for your future psychology classes. So now let's just see how to find them. So here's some screenshots from Google Scholar. Google Scholar is going to be your second best friend. So someone give me a topic. This is where you can find academic journal articles. What's an interesting psychology topic we should look up? None of you are sure. Yeah. Sorry? Cognition. Maybe something more narrow?
Of fish.
The cognition of fish. That'd be dancing with the line of what I'd accept, but you know what? What the hell? I'm gonna give you. Let's try cognition of fish. Social cognition. Wow, I didn't think they were all that social, but hey, okay. So you find the article, click Cite, and then go to the APA one. Now, it should have this format, right? With like the number and then another number in brackets and then page numbers. If it isn't formatted like that, find something else. It might be a book chapter or a law review or something. Only use it if it has this format. And then you'll just paste it into here and make sure you change the format to be exactly like this. Google Scholar is good, but it's not perfect. In fact, there's a mistake in this one. Cognitive and sciences are not capitalized. In should be lowercase, but these should both be capitalized. So make sure it's the same as my example. You can also look up, like on Google, APA style or whatever, but you have all the information you need in the template. Okay. Any questions about this assignment? Should be pretty straightforward. So upload early, follow the template, no creativity, and you should be fine. So I'm going to say a bit about what these articles are. So here's an example of a paper in my area. which is moral psychology. So they'll all start off with an abstract, which is a quick summary of the whole paper. The first chunk is the introduction, which summarizes the prior studies that have been done on the same topic. There will be a methods section where they describe exactly what they did. The results section where they summarize what they found. So that's where you'll see the statistics, which again, you don't need to worry about because you're not yet trained in how to read. And there's the p-value, you at least know what that is. Under 0.05, that's what we want, just barely though. And then the discussion is where the results are integrated with prior studies. So sort of the meaning of the results is explained. These studies are the bread and butter of an academic career. When you're a PhD student or a professor, your goal is to publish lots of these. You may have heard the phrase publish or perish. That's kind of the mantra of academics, and they try to get lots of these published. And it's kind of an arduous process because you have to write this up, which is, in itself a challenge. Then you have to submit it to a journal. The journal will then select 2 or 3 reviewers to read it and see if it's good. And the readers will be like other experts in that area. And then they decide whether or not to accept the article. And there's kind of a hierarchy, right? So this is in Cognition, which is a pretty good journal. So usually what you do is you aim for like the best journal. Most of the time you get rejected from there and you kind of move your way down until you get the article published. So that's what these are. And most of the sources in the book, when you see like in brackets names, most of those sources are things like this, journal articles that have been peer reviewed and then published. So as I said, I'm going to use this as a springboard to talk about what an education in psychology looks like. And as always, go ahead.
I was just wondering, what's the limitations? Because you had said that's kind of even the one on fish was kind of like a stretch. So what is the limitation?
Here's one easy thing to do. If the journal has the word psychology in it, or cognition, or some psyche word, then it's probably fine. Oh. I'm not going to be super picky about it, but I mean, if you give me a journal article on almonds or something, okay, no.
Sure.
Yeah. Okay, so some of this will be reviewed for most of you, but step 1 is to get a BA or a BSc, so Bachelor of Arts or Bachelor of Science in psychology, which typically takes 4 years, although increasingly people are taking longer, but in theory it should be 4 years. So if you major in psychology, you'll have to take some statistics and research methods, but mostly upper level courses like social psychology or cognitive psychology or stuff like that. And if you're planning on going on into graduate school, you should try to get a honors degree. So this university, like most, has an honors program. And when you're in the honors program, you do an honors thesis. So you'll do a little study, you'll write a little report on it, kind of like a journal article, maybe a bit shorter. Which is good practice for the kind of thing you'll be doing in graduate school. So if you want to go into graduate school, it's recommended that you try for this. The next step is to get a master's or Master of Arts or Master of Science. That takes, depends on the kind of program, 1 to 3 years. 2 is pretty common. And then a PhD, which is 3 to 5 years. So that's kind of the standard path. Now, within the master's and PhD, there's 2 kinds of paths you can take. One is the clinical or counseling route, where you'll still have to do some research, particularly if you're getting a PhD, but the emphasis is more on getting experience being a counselor or a clinician or whatever, helping people. So it's more applied. And if you're applying for a program like that, the big thing you want to get before is some kind of real-world experience, like suicide hotline, addiction center, some kind of practical psyche, thing. Research experience is good too, but you have to have some kind of applied experience. The other track, which is the one I'm on, is experimental psychology, where we don't care about helping people. We just want to understand how the mind works. So social psychology, cognitive psychology, evolutionary psychology, those would all be in the experimental track. And for both of these, you are expected to produce some kind of original research. So if it's a master's, then it's a master's thesis, which is 1 or 2 studies, kind of more narrow. If it's a PhD, you want around 5 studies, maybe a little less, a little more, kind of a larger original contribution to an area of study. So then you'll do what's called a PhD dissertation. And the courses are no longer what matters. This is the most difficult transition when you go to an MA or PhD. You take some courses, but no one really cares. I mean, you're expected to get A's, but it's just like, well, yeah, of course, you're in graduate school, of course you're getting A's. What matters is doing original research when you're in this track. And then while you're there, you're also expected to be presenting your work at conferences. That's another big thing you do. There's academic conferences where grad students and professors will present their results. Yes?
If you're wanting to go into honors, like what year should you, like in what year should you look at like trying to find like a supervisor and stuff for your like?
Yeah, I think you're expected to start by your 3rd year, so kind of end of 2nd year you
That's
start
when you
looking.
should start looking?
Start looking, I think so. Now double check the website. I didn't do my undergrad here. In my undergrad you started in your 4th year, but I'm pretty sure here you started.
Yeah, I'm pretty sure it's 3rd year. I
Yeah.
just didn't know like when you should probably.
Yeah. So yeah, that's the idea there. Yep.
So what's the difference between the arts program and the sciences program in psychology?
It mostly has to do with what other courses you're expected to take. The courses you're expected to take within psychology, my understanding is it's pretty similar, but you have to take more math and more science courses if you take the Bachelor of Science. My college didn't have a Bachelor of Science in psychology, so I'm less familiar with that, but that's my understanding. Yeah.
In like for like research related jobs, do they like look like would they pick someone with a Bachelor of Science over someone with a Bachelor of Arts when it comes to like if you wanting to go into more scientific research?
It really depends what kind of area you're in. I'm not sure it matters very much. I mean, I'm in a lab that's more sciencey. It's a neuroscience lab. I had a Bachelor of Arts and actually a Master's in Philosophy, and I still got in. So stuff like that, it really varies by supervisor. There's a lot of kind of randomness built into that. But yeah, any other questions about this general picture of what an education in psychology looks like? Okay. Now, I'm going to add something else, and I feel like the department might be mad if I say this, but I think it's good to give this information to students as well. There are pros and cons to getting a degree in psychology, and I'm going to say it like this. As far as I'm concerned, there are 3 possible reasons you could be looking to get a bachelor's degree in something. One is it's just interesting. Maybe you're spoiled and your parents are paying for this anyways, and you think, oh, I want like a more broad view of the world. I want to be more well informed and a more sophisticated consumer of information, something like that. Some of you may want to go from a bachelor's degree to grad school, kind of like I was talking about, and then to a job, right? You may want to go into graduate school, or maybe like law school or medical school, something other than just an undergraduate degree, and use that as a basis to get a job. And some of you may want to go straight from a bachelor's to a job. If your goal is number 1, in my view, a little biased, psychology is one of the best. Very interdisciplinary field, you will learn a lot, and it will enrich your understanding of the world. For number 2, again, I think psychology is excellent. There's lots of different paths you can take if you're willing to put in the work to get into graduate school and to do well while there. Oh, and I forgot to mention, I'll just add, since we're in this kind of an info session, when you're applying to graduate school, so obviously GPA matters, You will have to get 3 letters of recommendation from professors. So it's good to build relationships with professors, especially in your 3rd and 4th year, where the classes are smaller. So that's another thing to keep in mind. That's harder in a college or university that's this big, but that's a good thing to think about. You'll have to write a statement of kind of what you're interested in doing while in graduate school. And for some programs, you have to take the GRE, which is a standardized test, kind of like, some argue it's basically an IQ test without them calling it that, but it's kind of reading, writing, vocabulary, and mathematics. So for some programs, you have to take that, although a lot of schools are getting rid of it, so you may not have to. But again, for this path, psychology I think is very good. Lots of opportunities. You know, counselors and clinical psychologists typically make $100 to $200 an hour. So, yeah. Exactly. So lots of good paths on this one. For number 3, psychology is not the best. Maybe don't tell the department I said that, but if you want specific, marketable skills that'll get you a job right after your BA, I mean, psychology is not totally useless. I mean, I do know a guy who worked in addictions and mental health. He worked there for a few years, and once he got his BA, they were like, okay, you're a supervisor now. Like, you have experience, everyone else here has experience, you're obviously more qualified to work with people than people who have literally nothing. And it's also true that there are a lot of jobs that just want you to have a bachelor's in something, right? Because if you have a bachelor's, even if they don't know that you have specific marketable skills, they know that you're smart and that you're hardworking and committed enough to finish a degree. But as far as specific marketable skills, psychology is not necessarily the best. Any last questions about this kind of stuff? Okay. So you have the book, and we're starting chapter 5, talking about sensation and perception. And I'm going to call these 1 and 2 because you can think of them as sort of the 2 steps in coming to see something. So obviously what we're talking about here is the 5 senses. I mean, there's actually some others, but mostly the 5 big ones. Sensation is just detecting the incoming signals, right? Just that kind of step 1. So the light goes into the eyes, they send a signal to the brain, maybe some early processing, that's sensation. Just that initial detection that something's going on. So let's say I'm looking at a red apple. Okay, red light bounces off the apple into my eye. Okay, and it goes to the occipital cortex. That's the raw kind of sense data. Perception is putting that information together. And, oh, it's round. Oh, it's an apple. And it's making sense of the incoming data. This is not a sharp line. In the middle, it's kind of fuzzy. But in general, that's the distinction between them. The book has a nice picture of it. Kind of related to this is what I'm going to call step 1A, which is transduction. This is the first part of sensation for all of the senses. So transduction. This will definitely be on the test. I haven't made the test yet, but the chances of this being on the test are 100%. Very important concept. It's always, as I say, the first step. So transduction is the transformation of signals from the environment into neural signals. Transduction is the transformation of signals from the environment into neural signals. So this is very important because think about vision as an example. It's the one we're going to spend the most time on. There is no light in the brain, right? It's dark in there. So in order for the brain to process information about light, the light has to be turned into neural signals. So light hits cells at the back of the eye, like in the retina, and then those cells turn the light into neural signals. And the neural signals are then what is processed in the brain. The same is true for hearing, for touch, and for smell, and for taste. So that's crucial. That's always the first step. Or I shouldn't have. Any questions? I mean, this is pretty basic introductory stuff, but any questions about that? Okay. The book starts the chapter off with a discussion of synesthesia. The spelling is in the in the list of key terms. Synesthesia is when your senses get mixed up. I'm not going to spend too much time on this, but there's kind of spooky versions of this, like where you see certain colors and they trigger a certain smell, or you hear a certain kind of music and you have a visual experience. Probably the most common kind of synesthesia is for letters and numbers. So there's certain people for whom every letter and digit has a certain color, just instinctively. And they're not making it up, it's not something trick they're playing on themselves. There's some deep kind of crisscrossing of wires in the brain. So I knew someone who had this and she was very young. And so for her, I forget which was which, but let's say like 5 is blue and 1 is orange. And for her, that this connection was just absolutely deep. And she was in a math class, and the teacher said, flip to page 51. And she was like, oh, flip to page blue-orange. And the guy beside her was like, what the hell are you talking about? And she was like, no, whenever you think about 5, it's always blue, right? And he's like, I'm thinking about it, it's black. I don't know what you're talking about. So it turns out she had synesthesia and kind of assumed that everyone did. So like I said, I don't have too much to say to that. But that's what synesthesia is. Okay, here's a question. I've asked a similar question before, but there's a few new options here. So think about the 5 senses. So vision, hearing, touch, taste, and smell. So here's a little activity to do. Which would you give up in which order? So if some evil genie or something, I don't know, came up to you and said, you must give up one of the senses, which one are you giving up first? Just write them down, and that's number 1. And then number 2 is the one you'll give up next. And so on until you get to number 5. Number 5 is the one you're keeping till the end. A little activity to kind of think about the relative importance of these. I'll give like another 20 seconds to think about it. Okay. So let me just ask. All right, wrap up your conversation. Okay, I'm going to focus in on number 5. So think about which one you'll keep to the end. How many of you, number 5 is vision? Exactly, very typical class. So there's studies on this, that is what most people say. This is because we are primates. We are heavily reliant on vision. This is also reflected in the fact that vision takes up more of the brain than any of the other 4. Which makes sense given what we saw in chapter 3, right? The whole occipital lobe is for vision, but also parts of the temporal and parietal, right? Vision is a very greedy sense. But that's not true for all species. So if you're a bat, but if something is a bat, it's hearing that they most rely on, right, through echolocation. Some animals have much better senses of smell than we do. By the way, how many of you is smell number 5? How many of you is smell number 1? Right, it's a very, very common answer. Humans have very weak senses of smell. A very small amount of the brain is devoted to what's called olfactory processing, which is smell processing. So we're very heavily reliant on vision, and as a result, much more of the book is devoted to vision than taste or something. Okay. Next, we'll talk about different kinds of thresholds, in particular 2 kinds. The work on thresholds was done by people in an area called psychophysics. I won't test you on that term, but it's essentially looking at the relationship between physical properties of a stimulus, how bright the light is, how loud the sound is, or whatever, and our brain's reaction to that stimulus. And the first kind of threshold is the absolute threshold. The absolute threshold is the smallest amount of stimulus that you can detect 50% of the time. Smallest amount of stimulus that you can detect 50% of the time. Or I should say at least 50% of the time. So here's a little thought experiment to help you. Suppose you're sitting in a dark room and I have a special machine that can choose exactly how many photons of light to send. Right? Photons are particles of light. So I send 1 photon. Obviously, you're not going to notice. Right? It goes into your eye, you can. And then I send 2 photons, 3, etc. I slowly increase the amount of light until you can just barely see it. That is the absolute threshold. It's the smallest amount that you can just barely detect. The same would be true for sound, right? You slowly make the sound louder until you can just barely hear it. So for vision, the absolute threshold is very small. We can detect very small amounts of light. Okay, the other kind of threshold is the difference threshold. The absolute threshold is about whether you can see the stimulus at all. With the difference threshold, there's 2 stimuli, and it's not a question of whether you can see them, it's a question of whether you can tell that they're different. So, like 2 shades of red, and one is just very, very, very slightly darker. How much darker does it have to be before you can tell, those are 2 different colors? Again, in that case, very small. The difference threshold for color, very small for humans. Here's another example. Suppose you have to detect whether 2 weights are, if one of them is heavier. Right? You have to, there's a different threshold for that. So obviously if I gave you like a 30-pound weight and a 30.0001 pound weight, you're not going to get it right any more than chance. You're just going to guess. You have no way of detecting which one is heavier. By the way, another term for the difference threshold is just noticeable difference, which is pretty intuitive, right? It's the difference that you can just barely notice. And a related term in the book that you should know is Weber's law. I think I'm going to have to bring new felts next time. That's what it looks like to me. But yeah, W-E-B-E-R, Weber's law. And Weber's law essentially boils down to this. The difference threshold is always in terms of percentage of difference, or fraction of difference. never absolute amount. So let's go back to the weight example. Suppose I gave you a 60-pound weight and a 59-pound weight. Can you tell which one's heavier? No, right? Oh, they're both heavy. You can't tell. That's 1 pound of difference. But if I gave you a 1-pound weight and a 2-pound weight, you would easily be able to tell the difference. Even though the amount of difference is the same, it's 1 pound. But the fraction of difference is much bigger in the second case, right? It's double the weight, right? So that's what Weber's law states. For all the various senses, there is a specific fraction of difference which decides whether you can tell the difference or not. It's not in terms of amount. Any questions about that? Okay. Next, signal detection theory. This is about kind of hits and misses and other kinds of errors. It's about how we detect things. Let's see if the green is better. So let's say you're in that situation that I described where there's like a light that's sending like a very small amount of light at a time. And you have to decide, did you see something or not? The book has a helpful table here, which I'm going to reproduce. Actually, I'll have to make it lower. So there's two things going on. Is there a stimulus? So in this case, was there a light or not? So no and yes. So here it's yes, here it's no. And then there's your decision. Right? Was there something there or not? No, yes. So if there was a light shown to you, and you say that there was a light there, well then you're here, well then you're good, right? You made the right decision. You correctly detected that there was light there. If there's no light, and you say, nothing there, again, well then you made the right decision. So these boxes are two types of errors that you can make. And the term for these, this is a different term from the book, but it will help you in future classes, is false negative for this box, and false positive for this box. The easy way to remember this is the falseness is always about what you say. The world isn't false. Like, that doesn't make any sense. What can be false is what you say. So let's say that they flash a light, so there is a stimulus present, but you say, nothing there. That's a false negative. You said there's nothing there, but there was something there. You definitely do not need to know this term, but in statistics, this is called a type 2 error. That may help you in a future class. In this case, there's no light there, but your eye plays a trick on you or something, you go, oh, no, I think there's a light there. Okay, in that case, you made a false positive. Or the statistics term is type 1 error. Because again, only your decision can, or what you say can be false. So the positive has to do with what you said, and the false means, yeah, you're wrong. So that's the basic, the core of signal detection theory. And I'll note, this has application not just for perception, but for testing scientific theories. So this next bit, you don't need to worry about, but it may come up in a later class. So forget about perception. Think about a scientific hypothesis, right? So your hypothesis could be true or false, right? Your guess about the world. So let's say you have a new drug and you think it works. So yes would be, yeah, the drug works. No would be, nope, the drug's a piece of trash. You can make these errors in that case too. So you do a study and you conclude the drug does not work. You say, nope, not enough evidence to conclude the drug works, but you may be making a false negative. The drug may actually work, maybe you just didn't have a big enough sample to detect the effect or whatever. But conversely, you may conclude the drug does work, but really it's a false positive. You just had some sampling error, or you did your statistics wrong, or there's different ways it can happen, but for whatever reason you made a false positive. And this one difficult issue to think about is it's kind of up to us how strict we are in science. So we can just be super strict, like we're only going to believe things if there's 100% conclusive evidence for something. But if we make it that strict, then we're going to have a lot of false negatives. We're going to reject a lot of hypotheses that are actually true, but we were just being too strict. But obviously, if we're too loose and we just kind of believe anything, even with a tiny bit of evidence, well then we're going to have a lot of false positives. We're going to believe a lot of bullshit theories because we weren't strict enough with whether or not to accept theories. Anyways, that's just a bit of an aside, but it's rooted in the same idea of signal detection theory. Any questions about signal detection theory?
So, this means the error.
Sorry?
This means the error.
Yeah, it's about different types of errors that you can make when trying to detect something.
And this is too bad for now.
Say again, sorry?
If I'm not guessing, we will these two.
Yeah, as long as, I forget if I put false negative and false positive in the key terms. So double check those, but yeah, I would say I know what a false positive is and a false negative is. But don't worry about it in the context of scientific hypothesis testing, just worry about it in the case of perception. So, is there a light there or not? Yeah, and know what the two errors mean in that context. Okay, any other questions before we move on? Okay. The next section talks about sensory adaptation or habituation. And these are used synonymously in the book. This is just getting used to something, right? So your reaction to a stimulus in the environment tends to go down. Here's an example. How many of you have a phone in your pocket? You're not in trouble, like you're just... Okay. If you can, well, first of all, just notice how little attention you're paying to it before I said anything. It's in your pocket, it's pressing against your leg, but you probably gave it no thought. If you take it out and put it in the other pocket, now you're kind of more aware of it, right? Like it's like, oh, it feels like it's like pressing more against my leg. You're kind of more aware of its presence. That's because while it was on the other leg, it had been there for a while and you kind of got used to it, started ignoring it. And if you leave it in the other pocket for a while, the same thing will happen. You will become habituated to or adapted to the stimulus. Pretty simple idea. Okay. Next, we're going to talk about vision. So this is where I have the first video of the day. So the book devotes the largest percentage of time to vision. I'm hoping to finish vision today. There's a chance I don't have time to, but we will try. And I'm going to start with a video. This is a video of someone who wore upside-down goggles for, I think, a week. So when you wear these, like, light gets kind of inverted, so the whole world is flipped upside down. And he wanted to see if he could learn to ride a bike with these goggles on after a week. So we're going to watch that, and then we're going to discuss it. Don't care about his Patreon. Okay. So pretty amazing that within just a week of experience, he was able to navigate the world successfully, and that subjectively, the world seemed to kind of be right after a while. Yes?
Is it true, just like a urban legend, that like some people's brains don't flip their vision and they do perceive the world upside down?
Oh, I've never heard that. Now you're kind of spoiling the punchline, but the answer is I have no idea. Now, as she has sort of spoiled, the punchline, I had a really clever line. I was going to say, oh, some of you are wondering how he did that. And then the punchline was the same way you did. Because as this person notes, the eyes actually perceive the world upside down. Objects up here, the light goes down there. Objects down here, the light goes up there. And the same is true with the pattern of activation in the cortex. It's all upside down. And you might think, well, how does the brain have to like flip the image over or something? And that rests on a mistake about how the brain works. There's a very tempting view according to which the brain has like a little picture in there, or a theater where the movie plays. And that's a very misleading picture. What the brain's processing is for guiding your behavior adaptively. That's what visual perception is for. And as long as you can habituate yourself to learning to use the incoming information to guide your behavior correctly, it kind of feels right then. It's not as though there's an image in there that has to be flipped over. That's a very misleading view of how it works. Okay, so now we're going to get to see more of my wonderful art. So I'm going to draw a picture of an eye, and we're going to go through the main parts. So this may be a review from bio classes, but the parts of the eye are testable. So the first layer that we've talked about is the cornea, which is just the clear layer on the top of the kind of front of the eye. So it's see-through. The colored part, so if you have brown eyes, the brown part. If you have blue eyes, the blue part. For a long time, I didn't know what color my eyes were. I was kind of like debating between blue and green. They were really indecisive. A while ago, I figured out there was an app. You just take a picture and then you can click on any pixel and see what color it is. And apparently they are slightly more green. So it turns out I had green eyes all along. But whatever the colored part is, that's the iris. And then in the middle of the iris is the pupil, the black, which is just like a hole in the iris through which the light goes. The iris can constrict or dilate to make the pupil larger or smaller. That's kind of its job. So light comes in through the pupil, and then it goes through something called the lens, which is behind the pupil. If you have bad vision and require glasses, blame your lens. Most conditions like that result because the lens is the wrong shape. And then the lens focuses the light onto the retina, which is the back of the eye, where all the light-sensitive cells are. Again, that's why things are upside down as far as how they're perceived, because the light comes up. So if there's like a rock here, it's going to go up and activate stuff up here. I'll note in passing, so the lens is also what is cut, or parts that are shaved off, in LASIK surgery. How many of you had LASIK surgery? I guess most of you are probably too young, so one of you or two of you. So I had it done in my early 20s, strongly recommended. LASIK has the David stamp of approval. It's very painless, like it's very mild. I've had mosquito bites that hurt way more than LASIK did. It's just a strange experience because they peel back about a third of the cornea to get a clearer shot at the lens, and you see that. You're just like there. And then when they shoot the laser, you feel no pain because the lens doesn't have like pain detectors, but you can smell it. You smell your own burning flesh. So maybe this isn't as good of an advertisement as I hoped. I found it, and the recovery is super quick too. They just slap the cornea back on, and it heals like instantly. So, anyways, this was supposed to be an advertisement, but it may not be. Okay, so that's those parts. Now, the back of the eyes, as I say, the retina. The retina is where the light-sensitive cells are. And in the middle is a kind of tight cluster of light-sensitive cells called the fovea. The fovea is where you can see in the most detail. And if you want to know how big it is, if you hold your thumb out at arm's length, the top kind of part of your thumb, like kind of the fingernail part, that's about how big the fovea is. That's where you can see in the most detail. You might think, no man, I can see more detail than that. And that's actually kind of an illusion. I'm gonna come back to that actually in a second, but like if you're reading something, that's where you can see, as I say, in the most detail. Part of the reason you feel that you can see in detail more than that is your eyes are constantly saccading around, giving you more detail. Now, before I can say more detail about that, I have to explain there's two types of photosensitive cells, or light-sensitive cells. Rods and cones. Rods can only detect black and white. Cones can detect color. That's the main difference. And the cones are tightly clustered around the fovea and kind of what's called the parafovea, which you don't need to know, the kind of area around the fovea. Towards the outside of your vision, it's all rods. And again, many of you are thinking right now, no way, bro, I can see in color all the way to the edge. And look, I feel like that too. When I look ahead, I feel like I'm seeing color over here, but we are all wrong. That's an illusion. It's because you can kind of remember seeing color over here, and your eyes are bouncing around, kind of telling you what's there. But there are no color-sensitive cells out here. And we're going to prove this with a little demonstration. So I need 2 volunteers. That was fast. Okay, sure. Our perception of color is more narrow than it sometimes appears. Oh, and by the way, easy way to remember which is which: cone, see, color. Easy way to remember. Okay. Another spooky thing. Now this one's harder to demonstrate in class, but below the fovea is a place called the blind spot. So the rods and cones have axons that go out from them, and they all meet at this place where the optic nerve begins. But at that spot, there are no photoreceptors. There's no rods, no cones. So each eye has a blank space right in the middle. Now normally, both eyes are open, whatever, the other eye can compensate for that. But if you close one eye, you won't be able to see it, but there is actually a blank space in the middle, and your brain just fills in whatever's around there. And if you want to prove it to yourself, go home and look up something like this. If you look up blind spot, activity, whatever, there's one in the book. I think it's not a very good one. It'll tell you to close one eye and kind of be arm's length from the picture and kind of move around a little bit and stare at the cross. And if you do it from it won't work from here, but if you stare from the right distance, the black dot will disappear. It's very spooky. It'll just go white because the brain just fills in whatever's around there, and that corresponds to this spot where the axons are. Yes?
Is that why we don't really notice our nose?
No, it's not really that, because the nose would kind of be more in the periphery. This is close to the middle. Good question, though. Okay. Any other questions about the eye stuff? Oh, no.
Sorry, just for the black spot, can you give like just a little definition?
Yeah, the definition would just be, it is the place where the optic nerve begins. And it's the spot in the middle of your vision where you can't see. Okay. I'll briefly say something too about the evolution of the eye. The book has a discussion of photopigments, which are the molecules in the axons that detect light. Yes, sir.
I'm not understanding quite. So the brain just guesses what's there? Like it just fills in whatever information?
It fills in whatever's around it. So in this case, around the dot is white.
Yeah.
So it just goes, all right, more of that. Yeah. And again, if you can try it at home, you'll see it just fills it right in once you get to the right place. So if you close one eye, your brain's doing this right now. somewhere in there, it's just filling it in. Okay, so like I said though, so photopigments are what allow the eye to see. And this comes up sometimes in discussions of evolution, because some people say, oh, the eye couldn't possibly have evolved. But we actually have a pretty good understanding of the steps by which the eye evolved. Darwin, in The Origin of Species, laid out a sketch of how the eye could have evolved, and it's been largely confirmed. There are examples in more primitive species of the various steps that would have occurred. And once you see it, it's actually quite intuitive. So let's say there's a very simple, organism. The first mutation would have just been put photopigments here. Just have some cells that are light sensitive. That's step one. So kind of just on the surface here, just some light sensitive cells. And that's adaptive. That could be selected for by natural selection all on its own. This would have first evolved under water, and it's good to know where there's light or not. You can know that up is that way, because the sun, or if it goes black all of a sudden, that means something's above you, which could be bad. So all on its own, that is adaptive and could be selected for. But then, any incremental change that makes that surface dip a little bit is also going to be adaptive. Because if there's a dip, now you can know direction. If the light's coming from, hits this part, that means the light's coming from here. If it hits this part, that means it's coming from here, and so on. So a dip gives you more capacity to distinguish where things are coming from. And through incremental changes, you can get more of that, right? Just incremental dips. And there's some organisms that have basically that. And then there can be further selection pressure to have that gap start to close. Again, this is all step by step, because as it closes, you get even better detailed view of the outside. And you can probably see where this is going. Eventually, if you have the gap kind of close around a pinhole, I mean, there's some cameras like it's like a pinhole camera, you get the most detailed view of where objects are coming from. And you're almost home free at that point. Once you have it close up, and then if you have any kind of clear thing you can put in the middle, then you'll get like the most detail. So this will not be tested, but kind of an interesting bit of history of how the eye would have originally developed. The book then talks a bit about dark adaptation, which just means that when it's dark, your eyes slowly get better at detecting detail. Or not detail, but just better at seeing in the dark. Especially the rods. The rods are very good at seeing in the dark. One little practical tip from this, if you're looking for something in the dark, it's good if you look slightly away from it. Normally looking right at it is good because that's where all the cones are and you can see in detail, but if you look slightly away from it in the dark, you will actually be able to see it better because of more rods. Yes?
So like, if you spend more time in the dark, maybe be able to like slowly start in the dark.
Yes. Or, so to be clear, I mean like over the course of, what is the book say? I think it's like 30 minutes. Over the course of like 30 minutes. It plateaus there. So no, if you spend your life in darkness, well actually, you probably would get better at it, but not because the eyes are literally more sensitive, but you probably get more used to sort of cues to it. But that's kind of different from the sensitivity of the rods, which is more what this is about. But good question. Any other questions? Okay. I'm optimistic that we just might finish vision. The next part is on color vision. So black and white is kind of boring. The world would kind of suck if it was all black and white. Humans have excellent color vision. And there are two historically important theories of color vision, both of which you should know. Color vision is not my area of specialization at all, but there is apparently a new theory that tries to integrate the two views. But I'll start with the two historical views. The two historically influential views are the trichromatic theory and the opponent process theory. So the trichromatic one is the simple one. It says that there are 3 kinds of cones, 3 kinds of color detectors. Red, blue, green. So that's easy to remember, just those 3. And then those 3 combine in different ways to give you the different colors that you can see. This goes back to Young and Helmholtz. That's sometimes called the Young-Helmholtz theory, but you don't need to know that. The other view postulates that there are 3 pairs of opponent processes. And by opponent process, I mean, this is kind of a goofy way of putting it, but there's 2 processes that are fighting each other. And if one gets too active, the other wants to fight back. You can think of it like that. So one is red-green. So if you look at red for too long, you're like, red, red, red. The red gets active, and the green's like, hey man, that's not nice, and then wants to push back. And so the 3 pairs are red-green, blue-yellow, and black-white. Those are the 3 opponent processes. And there's definitely something to this latter view, and one way to see this is an American flag after image. So I don't know, have any of you seen this before? The American flag after image? Not too many. So hopefully it works for at least some of us. This one's a bit shorter. I wish there was one that was a bit longer, but we'll try this. So there's going to be an image of a reverse American flag, and there's going to be a red dot in the middle. Just stare at the red dot, and once the video's over, you can just look at the wall or something. And you should see a kind of fuzzy American flag. Okay, how many of you saw the after image? 2/3 or so. So these are freely available online. It doesn't work every single time, but feel free to try that again. But it should be pretty clear why this supports the opponent process theory, right? So if one half of the opponent process, you look at it too long, it's overactive, and the other one pushes back. There are lots of opponent processes in psychology. One that we won't talk about in this course is happiness. There's a theory called the hedonic treadmill theory. Lots of evidence for it. It's a kind of depressing finding, but the basic idea is whether your life goes really well or really badly, you kind of get used to it, right? So life could get really good, like you get a big promotion at work, you move to a new house, whatever. You're really happy for like a few weeks about it. And then you're back to your normal, neurotic, stressed self. There are very few things that permanently change your happiness. I mean, I guess the good news is the same is true for bad things. Quadriplegics, when they're first paralyzed, are very depressed. Not surprising. After a few years, basically back to normal. So that's another example of kind of an opponent process where things tend to kind of go back to homeostasis, or kind of a middle place. So sorry if that depresses you, but there's one other example of an opponent process. So there's clearly some truth to this, but the support for the trichromatic theory is if you just look inside the eye, there are only 3 kinds of cones: red, green, blue. So there's a view that I don't know much about called the dual process view, which says that both are operative. At the level of the cones, the trichromatic view is right. There are only 3. But they can combine in an interesting way to produce opponent process mechanisms. So the way it works is that the red and green cones, if they're active together in the right way, that can produce the yellow half of the blue-yellow opponent process. But the red and green together are opponent processes just with each other. So I don't have much to add to that. As I say, that's not my area of expertise, but that's the dual process view. Okay, before moving on to feature detectors, I have another video to show that kind of sums up some of the stuff we've said, and then I'll close off with feature detectors. Okay, I'll stop the video there. So I have a similar picture here. So as he said, information from this half of the visual spectrum goes to this hemisphere, information from this half goes to the other hemisphere. That's review from the prior chapter. And as he also said, it goes through the thalamus. The thalamus is what redirects sensory information to other hemisphere, or to the cortex. So, there's only 3 minutes left. Let's just stop there. Reminder that the review is due on Monday. I will see you all on Thursday.